This video will cover a brief introduction to polymers, their uses, and an overview of how biodegradable polymers break down. Polymers are large macromolecules made up of many small repeating monomer units joined together in a chain. For example, ethene can undergo polymerization to form polyethylene. They can be split into two main categories: natural polymers such as DNA, proteins, and polysaccharides, and synthetic polymers such as polyvinyl chloride, PVC, or polyethylene, which people use every day. Polymers are involved in almost all products that you can purchase today, whether forming the item itself or as part of its packaging and transport. They're versatile, durable, and useful in a whole range of materials due to their diverse properties which depend on the monomers that the polymer is made from. For example, they can form rigid plastic bottles made from polyethylene terephthalate, PET, or polymers can be more flexible in the case of plastic carrier bags made from polyethylene. Their uses are incredibly varied, from food containers, toothbrushes, and plastic straws to razor handles, nappies, and toys. However, whilst being chemically and physically inert is a useful feature in many applications, it has led to polymers having a devastating impact on the environment. Conventional polymers can last for hundreds of years after disposal, making them particularly inappropriate for applications in which plastics are used once and then thrown away. They're often difficult to break down, and packaging materials are usually contaminated by food or other substances, making physical recycling of these polymers impractical and expensive. Instead, researchers are developing biodegradable polymers to reduce their long-term environmental impact, especially for single-use plastics. Biodegradable polymers are materials that are designed to completely degrade when exposed to microorganisms, aerobic and/or anaerobic processes. Polymer chains can be broken down by enzymatic action of microorganisms such as bacteria, fungi, and algae. The excreted enzymes split long polymer chains into shorter chain fragments, which are transported by tunnel proteins in the cell wall into the microorganism, where they can be metabolized. Polymers can also be degraded non-enzymatically by chemical hydrolysis. For example, in polyesters, water attacks the ester linkage, splitting it into smaller molecules. Many natural biodegradable polymers are formed by plants using atmospheric CO2. For example, glucose monomers produced by photosynthesis join together to form cellulose. These polymers are naturally recycled via biological processes and biodegrade to form carbon dioxide, methane, water, minerals, and new biomass. The biodegradation of polymers is dependent on their characteristics, including chemical structure and molecular weight, as well as the environmental conditions of their surroundings. These include both biotic living and abiotic non-living factors. Polymers with hydrophilic groups, amino, hydroxyl, carboxyl, and amide groups, biodegrade more easily under certain humidity, and linear polymers are more easily biodegradable than branched and cross-linked polymers, as there is a larger surface area available for enzymes to work on. When biodegradable polymers are exposed to abiotic conditions including weather, humidity, different pH, and temperature, their macromolecular structure can be weakened, helping to initiate biodegradation. This is where biotic factors come in. There are three main biodegradable mechanisms as a result of biotic involvement. Physical biodegradation, where polymers are eroded by microorganisms and then destroyed due to cell growth. There's also chemical biodegradation caused by the direct action of biological enzymes secreted by microorganisms. This occurs in two steps. First, extracellular enzymes act on the surface of the material, breaking the polymer chains and generating smaller molecules such as carboxylic acids or esters. Next, these small compounds are assimilated by microorganisms and transformed into biomass and bioenergy, until final products of carbon dioxide or methane are obtained. Lastly, biodegradable polymers can be degraded by a mixture of biophysical and biochemical mechanisms, as microorganisms interact with polymers to form new macromolecules. The right levels of humidity are required in order for biodegradation to occur, as water is essential in the activity of microorganisms. This is why it's a major issue when biodegradable polymers are placed in dry landfill sites. The lack of moisture means they are unable to degrade effectively, and so will persist for many years longer than designed. One of the greatest contributors to plastic waste in landfill is polyethylene terephthalate, PET, with almost 70 million tonnes manufactured annually worldwide for use in plastic bottles, other packaging, and textiles. It is resistant to natural degradation, lasting up to 450 years in the environment. PET can be depolymerized to its constituents industrial via ester bond cleavage. However, this is rarely used on a large scale due to the high processing costs compared to production of virgin PET. Instead, products are mainly recycled mechanically, where new products are formed without significantly changing the chemical structure. Unfortunately, this results in a decrease in quality of the materials, thus reducing its value and limiting how many times it can be recycled. Often, they can only be recycled 2 or 3 times before quality decreases to the point where it can no longer be used. In recent years, the bacterium Idianella saccharensis was discovered, which is able to digest PET and use it as a major energy source. It secretes a PET digesting enzyme, PETase, which converts PET into mono-2-hydroxyethyl terephthalate, MHET, as well as trace amounts of bis-2-hydroxyethyl terephthalic acid, BHET, and terephthalic acid, TPA, as secondary products. Idianella saccharensis also secretes another enzyme, metase, which breaks MHET into 2 environmentally benign monomers, terephthalic acid and ethylene glycol. It's always important to consider the products of polymer degradation, as in some cases, the molecules that it breaks down into could be more environmentally damaging than the original polymer itself. The discovery of plastic digesting enzymes produced by evolving microorganisms provides promising starting points for future research and synthetic biology to break down man-made polymers more efficiently on an industrial scale. There is a huge variety of uses and future applications for biodegradable polymers from industry, sports and agriculture to medicine and pharmacy. New biodegradable polymers are being researched and developed for use as drug-releasing agents, absorbable sutures, and in tissue engineering. With plastic waste currently being generated at a shocking rate of almost 400 million tonnes per year, and with only 18% being recycled globally, now is a crucial time to start implementing biodegradable polymers into more everyday products. Additionally, more infrastructure must be developed for the disposal of biodegradable polymers in bioactive environments to enable them to break down in a reasonable timeframe. Overall, this plastic crisis is a complicated situation that requires urgent action to mitigate the impending environmental disaster posed by humanity's man-made plastic obsession. Thank you for watching.